Dr. Bharath, shall we start?
Good afternoon to all. Welcome to the afternoon session of today's FDP. And the speaker for this session is Dr. Bharath BR. He is a bioinformatician. He obtained his master's degree and PhD in bioinformatics from Kuempu University. Dr. Bharath has published around 26 research articles in reputed national and international journals and is identified as a technical reviewer for 15 international journals and is working as editorial board member for 3 international journals of high repute. Dr. Bharath is also a recipient of the Young Scientist Award from the Vellore Institute of Technology for his research in the areas of systems biology. Dr. Bharath is currently leading the computational biology team at Atrimed. His team is involved in the development of plant molecules database and screening of plant small molecules for desired activity using molecular modeling and simulations. Dr. Bharath, we are very happy to have you as one of our guest speakers for the FDP program, sir. So welcome, sir. And it is over to you for starting the session.
Yeah, thank you. Thank you very much, Salma ji, for your nice introduction. And it is immense a great privilege and honor for me to discuss something which I know with you all, since it is, to my understanding, it's a FDP, where we have our own faculties, and I'm very happy to tell that I was also a faculty for 10 years, and from past 2 years I have joined a industry. And at the outset, I would like to thank organizing committee of this FDP for giving me an opportunity to share my experience with you people. And I would like to discuss regarding the topic which is most commonly used, particularly in the pharma industry or in agriculture industry, food industry. So the major emphasis here is how can we fasten the process and cut the cost of research, because nowadays cost is what exactly is very important. So when it comes to the cost, time is the first cost for us. So how can we cut short the cost in terms of time as well as money? We are going to discuss in this. So I want to mention one thing here. You can stop me at any point of time by raising the hand, so that it will be my privilege to address whatever the questions or doubts you have in between. So please don't hesitate or don't feel bad to stop me anywhere in between the discussion. Shall I start, sir?
Yeah, please.
Yeah. Hope you all can see the screen here.
Yes, sir, it is seen.
Thank you. So firstly, I would like to introduce our organization. We at Atrimed are involved in plant-based research. Here we are working on several diseases, specifically focusing on autoimmune disorders. As we all know, plants are once again an integral part of our life. So how can we take a clue from these plants and identify potential therapeutic interventions to resolve some of the difficulties or problems, or particularly our healthcare issues, whatever we have in a society. So that is what exactly our major emphasis is. And here we have a database of around 4 lakh molecules in our organization. So all these molecules are being screened against different drug targets, and the hits will be identified, and these identified hits will go for separation. As we are working on plant molecules, we go for the separation and purification, and these purified isolates will be investigated in vitro, in vivo, and even in clinical trials. So this is what exactly the standard pipeline we have with the traditional knowledge. So with this brief introduction, I would like to
sir, could you switch on your video, sir?
Sorry, whenever I share the screen presentation, my video will be automatically off.
Okay, sir. Then fine. We can continue.
Okay. Since I have not installed a app, I'm using a browser. I cannot. Okay. Sorry for
Okay,
the
sir.
inconvenience. Okay.
That's okay. That's okay.
Yeah.
Please continue,
So
sir.
yeah, sure. So firstly, we will discuss like what exactly the pipeline we have in the process called as drug discovery or drug development. Because as I have mentioned earlier, time is also a cost. Cost doesn't mean to only a money. Okay, so as we all are aware, it's going to be a very lengthy and tedious process to come out with a new molecule or a novel investigational drug. So if it is a IND path, it's really a very big challenge, and this challenge has to be addressed very systematically. Otherwise, we'll be lost somewhere in between. So how efficiently we can address this particular challenge is up to the approaches or methods we are adopting in the process of drug development. So let's try to understand. It doesn't mean that you don't know. Just recall like what exactly the process or pipeline of drug discovery. As we know, this drug discovery pipeline can be divided broadly into two phases: preclinical phase and clinical phase. So in the preclinical phase, we have a chemistry part, pharmacology part, and authorization part. So to enter into the clinical trials, it requires a lot of confidence in proposing any particular product, investigational product for a clinical studies, and it requires really a substantial data. So here we have any scope to cut short this particular path? So this is where we have to think very seriously. When it comes to clinical part, definitely we don't have efficient methods to cut short the path. So we have a very minimum scope in our clinical studies. But whereas in preclinical stage, we really have ample opportunities to cut short our path by using bioinformatics or computer-aided drug discovery approaches. So we are majorly focusing on this particular preclinical study here, because this is once again cutting a cost of around 50%, if I'm not wrong. 50% of the cost which is involved in the process of drug discovery can be minimized if we use bioinformatics or computer-aided drug design approaches, and it minimizes minimum 3 to 4 years of time. So this is really a very good amount of time when it comes to the competition in a pharma industry. So if we are going to take a pace in the race of drug discovery for 4 years, really it's a good margin, I can say. So in this particular preclinical studies or phase, we have a chemistry part. So we also have a pharmacology part and authorization part. So authorization is once again especially focusing on IP. So how can we tackle the IP is once again going to be discussed in our coming slides. So let's go one by one. So when it comes to the cost of bringing any drug to the market, so it involves so much of expertise, it involves so much of time, efforts, and investment. So if we can sum up the cost which is involved in this process of drug discovery, the discovery phase itself costs us around 300 million US dollar. So further optimization is once again costing us more, like it is almost like 1/3 of drug discovery investment. So this is a zone where we can cut short the investment. This is why I have told we can reduce up to 50% of a cost of drug development process. So if you can sum up this first two rows, it amounts closely to 800 million US dollar, which is almost 40 to 45% of whatever the total cost of a drug discovery. So I'm not going to very specifically telling that it is going to involve 1930. Somewhere here and there we have some more or less cost, but when it comes to the drug discovery, it is really lengthy and cost consuming process. And with this cost, if you can buy a 380 Airbus, we can buy 4 Airbuses and we can really run the show. but it is not going to serve the purpose of healthcare industry, right? So with why I am sharing this slide or discussing with you is this is what the risk associated with drug discovery process. This is why industries are little skeptic and worried about taking IND projects. So without much delay, let's understand what is the mistake we are doing. So we all are adopting a Lord Anjaneya method. So please don't take it in any other sense. This is just for our understanding what exactly Lord Anjaneya method is. If you can just remember this incident where Lakshmana has gone sick and Lord Rama has asked Hanumanta or Lord Anjaneya to get Sanjeevani Mula. By then, Lord Anjaneya did not have any clue like how does Sanjeevani looks like, right? So this is what the stage or phase we are also in. When we are identifying some drug to any disease, we are in a Lord Anjaneya stage because we don't know which particular molecule in which plant is going to be a therapeutic option to cure one particular disease. So what we used to do earlier is we used to synthesize whatever the number of molecules we have, or else we used to isolate whatever the plant material available with, and we used to spend a lot of time in collecting, and we used to spend a lot of time on purifying them or synthesizing them and use screening them using high throughput screening. So this is how we have spent a lot of efforts in terms of time and money. Fortunately, Lord Anjaneya had a great powers and he could manage, but when it comes to the pharma company or a layman's like that, we cannot manage or tolerate such a situation. So we have to be smart here. So what exactly we can do is, if we have any kind of a vidya or any kind of a bless which can help us in identifying at least to some extent which particular plant or which particular plant molecule may be a real therapeutic intervention, then our purpose will be solved to some extent. So with this understanding, I would like to continue further to understand this involvement of CAD in preclinical investigation. So if I can split up this preclinical investigation, we have different stages here. Very first one is going to be a target discovery. So this involves identification of the disease and understanding of the disease. So first, when it comes to the industrial project or any projects, what exactly need of this project is very important. So that will be clearly spelled out by the seriousness of disease. So for that, we have to understand what exactly the disease is, what is the mortality is involved in, and what is the morbidity is involved in. And when it comes to the industrial setup, what is the market? So all these things are supposed to be clearly understood. And after that, okay, this is what the disease is and what may be the cause for this disease. So if you have to understand the cause of the disease, we have to know it at symptomatic level as well as at the causative level. So when it comes to the symptomatic level, it is a post-infection stage, and when it comes to the causative level, it is during the infection or prior to the infection. So knowing this at deeper level requires better intelligence. So this intelligence is going to be substituted by high-end computational power, which can really help us in identifying. So we are going to discuss about that. So next is if we understand a disease and identified one particular drug target, and now I have to screen which particular molecule is capable enough to inhibit this protein which is upregulated or which is involved in development of a disease. So this is once again a very crucial stage where we have to be very conscious in identifying the hit, because this hit is going to be taken for a next costliest effect called as lead optimization level. So we will understand what are the different methods we have and arms of bioinformatics we have to address these different stages of drug discovery. So we have these major arms of bioinformatics at different stages of drug discovery. Very first one is the genomics. Next thing is transcriptomics, proteomics, metabolomics, and systems biology. So with all these understanding, we will be definitely in a position to identify a potential drug target. So if we become successful in identifying the potential drug target, our 50% of problem is solved. So if we miss here, definitely we are going to end up with nothing. So these all arms are definitely helping us in identifying the drug target and even assessing the efficacy or safety of our therapeutic intervention. So let's understand one by one what is this genomics is. I'm pretty sure you all are heard of this genomics, and you may be much more familiar than me, but let me take a definition of this genomics. So genomics is a study of whole genomes of organisms and incorporates elements from genetics. And here it uses a combination of recombinant DNA, DNA sequencing methods, and bioinformatics to sequence, assemble, and analyze the structure and function of a genomes. So this is what the journey through the history of DNA sequencing. We all have come across this particular pathway. So firstly, in 1869, first DNA has got isolated, and now we are at the stage of next generation sequencing, where we also have a technology called as nanopore sequencing, where you can sequence a whole genome, okay, with a very low cost and with maximum efficiency. And nowadays, even Illumina sequencing has really saved a lot of time for us. So, and it has once again encouraged a sequencing approach. So just to understand how it has really encouraged us. So earlier in 2012, we were like with a very minimum sequences, and as we all know, in 2003, first human genome has got sequenced, and parallelly, a couple of model organisms genome has also got sequenced. But it was not an easy job for everyone to take up a genome projects, because it had involved a lot of efforts in terms of technology as well as cost. So gradually, newer sequencing strategies have got introduced. So it has reflected in improved number of genomes, and cost has dramatically come down. So now you can see in we can have a whole genome sequence with $1,000 cost. So this is how really sequencing approaches have helped us a lot. So just to put an example, during this pandemic situation, so COVID has hit the planet Earth probably in the month of November or October 2019, as per records. So by Jan 10th, the whole genome sequence of this COVID-19 was available in NCBI. So maximum people have taken 3 months of time in publishing a whole genome sequence of this COVID-19. Now we can understand the strength of technology, which has really helped us in identifying or in sequencing a whole genome of a virus, okay, in a very short span of time. So later on, as we all have witnessed, so many number of COVID-19 genome sequences have got deposited to the public domain databases. So it has become because of a genomics technology or advances in genomic technology. Then what these advancings has taken place? So earlier we had 10x genomics. Nowadays we are at the NovaSeq 6000 stage, and here we use these many techniques. Okay, and once we sequence, it is not done. The sequences are supposed to be assembled very carefully, and they are supposed to be mapped against the available reference sequences, and where exactly the insertion is, where is the deletion, or where exactly the single nucleotide polymorphism has taken place, what is the reason for this variant calling has to be understood, okay, and what are the CNVs and SVs have taken place, okay, and if the gene is existing in a genome, what is its expression level and at which particular phase it's getting expressed, or what is the function of this particular expressive gene. So to understand all this, okay, it is necessary to use some bioinformatics methods. So when does this bioinformatics has come into picture? When the amount of data started growing enormously, by then the need of this bioinformatics has come into picture. Why? Because when we have some amount of data or little amount of data, we can manage it well. But when it is going to be a vast or enormous amount of data, it requires very systematic organization of a data. So that is what exactly bioinformatics does. So we have primary, secondary, and tertiary databases, and these databases are playing a crucial role in acquiring the data through which is getting generated throughout the world and making it available throughout the world. So this is what the important role the databases are playing. So they are once again a main pillars of bioinformatics or biological data. So when we start getting this biological data, understanding of this biological data requires some critical analysis. So this critical analysis is aided by bioinformatics. So this is what the bioinformatics. So ultimately applications may be anything. It may be objective of this sequencing, may be whole genome sequencing and mapping it and understanding how it is different from earlier one, or how it is going to induce some other variations. So prospective and retrospective analysis can be definitely done, but it requires a bioinformatics tools. So researchers can choose a wide view to look at multiple samples or zoom in for in-depth analysis of a small specific area of interest on a whole genome. And this massive amount of DNA sequencing data can be generated very fastly and with very less cost never than earlier. And I'm not going to say that it cannot be reduced further. Yes, we still have options to reduce this cost involved in DNA sequencing. So with this understanding on genomics, we can move further. So let's take a note here, like we got to know that some gene which is novel in the particular patient, so that may be responsible for the development of disease. So if you have 4 to 5 genes which are seen freshly, or else which were not noticed earlier in a particular patient, then we can say that yeah, either 1 or 4 to 5 genes may be involved in this disease development. Further, it doesn't mean that if the gene is present, it is expressive in its nature. So as we all know, genomics is a comprehensive analysis of expression of any gene in a particular instance of time. So just because the presence of gene doesn't mean that it is expressive. So to understand that, the concept of transcriptomics has come into picture. So what exactly this transcriptomics is? So this is once again next stage of genomics, we can say. Here we can understand the expression of this gene at mRNA level or at expression level. So here once again we have 3 to 4 techniques like RNA sequencing, RNA microarray, or we can even call it as a DNA microarray, and expression sequence stacks, and we also have a technique called as SAGE, serial analysis of gene expression. So here you can understand with this, earlier we were using ESGs most prominently. Nowadays, after RNA sequencing, as the sequencing techniques have grown, we can see significant drop down in other techniques like microarray, SAGE, and expression sequence tag. But RNA microarray or DNA microarray is still a good approach, or approach which can give us a better confidence in understanding the gene expression. And SAGE is really a good method, but it has not taken a phase because it is a little bit tricky, as well as cost consuming. So that's what has really made researchers not to adopt this SAGE enthusiastically. So with this understanding, I'll go further in deep regarding what is the difference between this SAGE, DNA microarray and RNA sequencing, and what is the role of this bioinformatics here. So when it comes to the innovation, definitely we have to look into both the pros and cons of any technique so that we will be in a position to reach a logical end. So when it comes to SAGE, here I'll just focus much on in silico part. Here after the SAGE, we will come out with some tags. So we call them as once again a sequence tags or expression tags. This tags deconvolution data will be obtained, and we take a count of each and every tag, which particular expression tag is with high count. So looking at this tag, we can say, yes, the gene responsible for the production of this protein is having a more number of tags, so it is highly expressive. So counting the tag, we are going to come out with. So next, when it comes to DNA microarray, the label fragments will be bounded here to an ordered array of complementary oligonucleotides, and measurement of fluorescent intensity will be the key factor here. Okay, so as we all know, we are going to get a fluorescent chip, and the intensity of a light will be a measuring factor here. So fluorescence intensity will be measured using some image processing approaches, and here the intensity will be directly proportional to the expression of a gene. So this is how DNA microarray works, and how RNA sequencing is really advanced than this. Okay, so how it has taken a very good call, okay, or very good page in a research of transcriptomics. So the result obtained from this RNA sequencing, or data obtained from RNA sequencing, can be used to annotate where expressed genes are. Whether the gene is expressive or not is the first part. But in this sequencing, whether it is expressive or not, if it is expressing, what is the sequence of a RNA, that also can be understood, so that if there is any gross error in an expression, okay, or if there is any variation in expression, maybe due to one particular nucleotide or set of nucleotides in the genome, that can be very critically understood both at expression as well as at the splicing level. So this is what is missing in DNA microarray. If it is expressed, if it may be expressing, but due to some splicing error, our probes may not bound or hybridize this RNAs. So expression would be there, but it would be missing because of a splicing error. So that won't be there in this strategy. So it requires a lot of bioinformatics techniques, where we will have to align the sequences or assemblies which we obtain after sequencing, and annotating back them for a reference sequence, and where exactly the mutation or variation has seen can be identified. So most of the people discusses this methods, but it is very hard to decide which database we have to refer, right? So we have so many databases from leading databases in the world like NCBI, ENA, EBI, DDBJ, but which one to take and what kind of the data it is storing is very important. When it comes to GEO, that is Gene Expression Omnibus, which is offered by NCBI, it is going to cover, or it is storing information on microarray RNA sequence. So nowadays, GEO is also collecting the sequence information, as well as it covers a microarray information. So this is, in my understanding and experience, GEO data sets are, or series are the best to my knowledge, and I'm very satisfied with this. And when it comes to Array Express, ENA maintains this Array Express. Here solely microarray data is available, and it imports the data sets once again from the GEO, and it accepts the direct submissions even from the researchers around the world. So it takes the data or borrows the data from both GUI as well as direct submissions, so that the data which is available in microarray or ENA may be lost if you refer to NCBI. So I always recommend to look into both, whether it is available in ENA or whether it is available in NCBI. So it is better to check in both. Sorry. And you may ask me like, sir, anyhow the ENA is borrowing the data from NCBI. If we refer to ENA, it is more than sufficient. It covers both. Definitely not. Why? Because while borrowing, it try to avoid the redundancy. So if there is a small difference in similar type of data, that would be missed. So I strongly recommend you to go for both. So if you go through EBI, nothing wrong, because NCBI and EBI shares their information. Okay, so both are going to give you they exchange the information, and if you access through EBI or NCBI, you will be getting the same data. So sorry, I'm just skipping this. Likewise, we also have some databases like Gen Investigator, RefX, Non-Code, all these databases. Why I'm listing this is when we go for the curation of our data, some additional features are available here. Definitely, expression data is available in all these databases. Annotations and other features are available in different databases. So with this understanding on genomics as well as transcriptomics, now let's go to proteomics. So as we have discussed, if there is a gene, it may or may not express. If the gene has got expressed, it results with mRNA. So just because mRNA is expressed, it doesn't mean that it has to produce a protein, because as we have discussed, a small splicing variation may lead to the unsuccessful translation, right? So what researchers have thought is, there are some limitation in gene expression. Okay, so we will have to analyze or understand it in a still deeper way at a proteomic level. So let us take a case like we have understood a whole genome of one particular organism, that is COVID. Okay, during the COVID infection, people have expressed these many genes. So that was understood by means of RNA sequence analysis or microarray analysis. Nowadays, we are not using microarray analysis extensively, but we use RNA-seq. So through this, we will get to know, these are the inflammatory mRNAs which are responding during the COVID-19 infection, but how effectively they are ending up being up proteins. So to evaluate that, so we use proteomics strategies. So this proteomics is once again a comprehensive study of proteins which are expressed in a particular instance of time, and these proteins are involved in multiple cellular functions in association with co-expressive proteins. So this is what we have understood. So none of the proteins are active individually. They are going to be active or involved in some mechanism in collaboration or in coordination with some other protein. So which protein is regulated by which protein has to be understood. Otherwise, which protein is regulating another protein? If I stop this particular fellow, whether it is really serving the purpose or causing undesired effect. So that also has to be understood, right? So for that, this proteomic analysis is pretty much essential. So the proteome is the entire set of protein that is expressed or differentially expressed in an organism or system in a particular instance of time. When I say instance of time, it may be a disease state, or it may be a healthy state, or it may be a physiological state, or it may be a pathological state. So the proteins expressed in a pathological state need not to be same in a physiological state, right? That's why it has become a pathological state. So to understand this, we need this proteomic approaches. So here we will get to know which protein has upregulated or downregulated and involved in the development of disease. So what exactly this genome versus proteome is? So when it comes to technical aspects, essentially these genomes are static over a time. So very next minute genome will not change, right? So but proteome is very dynamic over time. When you are hunger, the expression is different. When you are full, your expression will be different. Likewise, non-location specific this genome is throughout 3 trillion or still more 3.2 trillion cells, we will be having this genetic material in chromosomes only, but these proteins are not having any location or specific location. They can be expressed at different locations in different cellular environment. So that is the complexity associated with proteome and even the specificity associated. Human genomes are mapped so far, but proteome is not mapped. Have you heard like this is what the proteome map of this cancer cell or of this particular human? No, we don't have any such a solid proof mapping for proteome, but for genome we have. That is the strength we have towards genome, and we have so far identified approximately 25,000 genes, but proteins are still inconclusive. In a last week, in a last month, we have come across a new cytokine called as IL-33, IL-41. Like this, we are coming out with newer protein, so that it is still inconclusive. And PCR is available to amplify this DNA to get it in a more number and go for studies. But we don't have any such a technique to quantitatively amplify your protein. So this is where difficulty associated with proteomics. So that's why whatever the data is available has to be very clearly maintained. So when it comes to the databases of these proteins, we have a well-known database called as UniProt. Here the data on almost all proteins of more than 1,800 species is available in this database. And we have a urine protein biomarker database, and here for a particular disease condition, what may be the biomarker, what is its expression, what is its differential expression, every information is available in this. And we also have a human kidney urine proteome project database. We have a Max Planck unified proteome database. Likewise, we have a couple of databases which are really giving us a valid and authenticated information. And here, whatever the data will be submitted to these databases will be reviewed carefully by the experts, and it will be made available to the public. So we can really trust that. Next is metabolomics. Just because your protein has got expressed, it may go fail in delivering its function. Why? Because in the process of biosynthesis of these proteins, okay, post-translational modification is very crucial. If a protein got synthesized but it fails to undergo a proper post-translational modification, it will be no longer available for its activity. So that activity may not be delivered efficiently by the protein, right? So instead of measuring the expression of this protein, how about measuring the expression of this metabolites? This is the call here. So people thought like, why can't we measure the metabolites? So as we know, these genomes consist of information on genes which are present or absent. Next level transcriptomes measures the active components of this genome. Next, in a proteome level, we measure the products of this gene expression. Okay. In metabolome, we measure the products of this reaction. So this is the highest level of understanding and accuracy we can attain at this point of time. Okay. So to understand it in a better way, okay, I would like to take another research article for a help. So this article has got published recently, and in this article, they have taken 3 different varieties of strawberry. Okay, so they had Benny Hop and L281, L84 variants, and they have focused on identifying the metabolites. Metabolites here are sugar, glucose, fructose, and total soluble sugars. So when it comes to the strawberry, this is an unripened stage, or we can still say it as a very tender state. And here it is bit grown, and here it is matured, and here it is ripened completely, right? So at each stage, okay, the taste of the strawberry is different. You can say, what is obvious in that? Definitely it happens, right? Yes, it happens because the gene which is expressive in a tender state, okay, may not be expressive in growth state. The genes which are expressive in ripened state may not be. So this is how the gene result in different metabolites. Okay, that's why the taste of this strawberry has changed over a period of time, right? So it has yielded different metabolites at different point of time. So let's look in terms of uh, metabolites. Okay, so when the particular variant, okay, is green in color, sucrose level was very low, right? So gradually it has got increased, right? So sucrose level was very high. So you can see in a Benny Hope, okay, in a Benny Hope, the sucrose level is very high, okay, because the gene which is involved in conversion of this basic reactant to the sucrose is very highly expressive. That's how the ripening, at the ripening stage, it has got almost 2 folds better sucrose. But when it comes to the glucose, okay, the gene which is involved in the conversion of a glucose, okay, was comparatively similar to L281, but it was bit different when you compare with the L84. So if you look at the image, okay, L84 pigmentation is not much differ, right? But its taste is going to be driven by these sugars, and we have a solid differences. So with this example, we can understand, I guess. So instead of understanding a gene's genomes, why can't we measure the products, end products? So in healthcare sector, I can say yes, we are measuring the blood glucose, right? Instead of measuring a PD4, which is once again involved in the development of diabetes. So likewise, we have different analytes. So I'm not going to say that this is the best, but in some instances, this sounds good compared to other techniques. So when we combine all these genes, genomes, proteomes, metabolomes, the complexity goes on increasing, because here we have a sequencing strategies. Next, we went on to understand its expression. Next, here in our 2D gel electrophoresis, we can understand, yes, this gene protein is expressive or not, but whether this expressed protein is active or not can be understood by ELISA, okay, and whether this expressed protein is resulted in a production of required or necessary metabolite or not can be understood using some analytical techniques like NMR, LCMS and all. So this is as the technology goes on offering you a best results, the complexity also rises, right? So ultimately all these are leading to the phenotype, right? So along with this, we also have a major factor called as environment, right? So this is what we actually miss. So we look into genes, we look into RNAs, we look into proteins, we look into metabolomes, okay? At different environment, they vary. If I am in a Bangalore, my expression are totally different when I move to Bangalore, right? So this is how environment is once again another factor. So to understand its influence, it is necessary to look all these in a systematic way. That is where the concept called as systems biology has come into picture. So this systems biology is very important. Okay, so why it is important, we will understand in a very short span of time, because this is my favorite area. If I start talking, you will have to stop me, because that really excites me to that level. So let me concise my thoughts here. So what is systems biology? So as per the NIH definition given in 2015, systems biology is an approach in biomedical research for the understanding of larger picture, okay, be it at the level of organism or tissue or at cellular level, by putting its pieces together. Okay, so by putting the molecular information together, how can we understand a whole organism or a tissue or a cell? this approach is called a systems biology approach. Okay, so it is in stark contrast to decades of reductionist biology, which involves taking the pieces apart. Okay, so in other terms, in a simplified way, we can define this systems biology as this area which aims at understanding biological complexity by unbiased measurements of as many as possible parameters without having any hypothesis, as it is understanding molecular complexity being outside proteomics, genomics. How better we understand is really our systems biology. So you can understand still in a better way here. As we go advanced in a omics technology, okay, the complexity goes high. Okay, but systems biology try to put all of them together and offers you a comprehensive analysis and give you a better confidence in any discovery research. So this method we are using with the polypharmacological approach in Atrimed. So it decreases the development time of drugs by half. Okay, that is by 50%, decreases the cost of development by 70% by reducing the trial and error approaches. Okay, so it can further reduce cost if coupled with traditional medicines also. Okay, because this is the point which I have purposely added. But why? Because when it comes to the traditional medicine or plant-based medicine, in m number of molecules versus n number of proteins is a proven hypothesis. Meantime, it can even be a m number of molecules against one protein, or one molecule against many proteins. So these are all the different approaches we have in our polypharmacology. Okay, so we will have to really thank to systems biology. So with this, we now have a clear picture on some complex diseases like COVID in a very short span of time. So with this, we will have to even look at the opportunities which are in the market. So if once we successfully identify the drug using all these approaches with the advent of systems biology, definitely we will have to go with a CAD, that is computer aided drug design. Here it is once again helping us to reduce the cost of drug development to minimum 30%. So this picture I have shared earlier, and this is bit extended part of the same. And here we have different approaches like ligand-based approach, structure-based approach, and system-based approaches. So when we will be having a clear information on a drug target, in the sense, if you know the structure of a drug target, by then we go for a structure-based drug design. Okay, so let's take an example of same COVID once again. So in January 2020, the genome of this COVID-19 has got released. Till then, we did not have any clue like which protein has to be targeted, what is the structure of the protein, what protein has to be stopped. Okay, we did not have any clue. By then, it is better to go for a ligand-based drug discovery where we will use some drugs with certain clinical rationale and we try to repurpose them against the required disease or disease of interest. So we adopt ligand-based drug discovery when we don't have a information over a protein or structure of protein. We definitely go for the structure-based drug design because this is comparatively accurate than ligand-based. So here, when we are available with the structure of this protein or target, we definitely go for structure-based drug design. So when we use both, okay, to arrive to some conclusion, we go for a system-based. Okay, so we will discuss about this. So if I just go on talking about this computer-aided drug design has got this much of opportunities, it has this trend, that trend, it may not sound good, right? So let's speak with respect to some research articles which have got published in highly reputed journals. Excuse me. So let's look into the paper we have here on the screen. And this particular paper has focused on design, synthesis, and molecular docking study of alpha triazolase isol. Sorry, I'm not a chemist. I cannot. It looks like a tongue twister for me. Okay, so here they are focusing on designing some derivatives for CD against CD22 ligands, right? So in this particular paper, what we have to focus is this is focusing on new non-hydrolyzable molecules, right? Right. So this synthesis has directed by CAD approach. So why I'm sharing this is if you go for the synthesis directly, you will end up with minimum 45 to 50 molecules. You have 50 molecules. You will go for screening of these 50 molecules. You will pick one, and the effort which you have put on 49 molecules goes waste. Otherwise, you will have to find some new target to that, and you will have to work on those, right? So to reduce this, what these people have done is they have done a screening first. They have identified a potential scaffold. After that, on that data, they have built or synthesized a molecule which has really given a good interaction with the target, and they have ended up with very successful results, right? So what exactly has to be the strategy we are supposed to have? So earlier when I was working with some chemists, most of the time chemists used to approach me saying that, Bharath, we have synthesized two series of molecules, man, and we need to do some biological activity for it. Could you please tell us what biological activity these molecules may give me? Otherwise, in some other worst cases, people used to complete biological activity and they used to come to me at the end asking for molecular docking to support this data. Do you think it is possible? It is very strange, right? So it is not an approach. So approach has to be like we should do rational based design of combinatorial library. We should do the screening first and go for the biological activity. This is what the proper pipeline, right? So it should not be like we go for the synthesis, some rational we will synthesize 100, 10, 100, 1000 molecules. We do the biological activity, and after that, to support that biological activity, we do the molecular docking studies and we go for the safety. So most of the studies will get fail here if you come by this route. If you go by the CAD enabled route, definitely we will have a very high success rate. So you can think like, yes, Bharath is a CAD guy and he is just checking, he is advocating towards CAD. No, I have a solid evidences, right? I'll discuss with you. So this is what the case where we have us. We get the data for this CAD from databases. Once again, protein data bank is a major database source we refer for the protein information. And if you have a protein structure information, fine. Otherwise, we will have to go for the homology modeling. And I'll not discuss much about this because this homology modeling itself is another topic to be discussed. So if I get an opportunity, definitely I'll discuss this topic in days to come. And how this data comes is it comes through electron density mapping or X-ray diffraction studies. Okay, so these structures are X-ray structures or X-ray diffraction structures elucidated by means of X-ray diffraction studies. So this crystallographic data provides us a protein structure. So what is the advent of bioinformatics is at any one particular corner of a world, if you elucidate the structure of protein, you are free to update your information or crystal structure into the database called as PDB, and you can get the crystal structure, okay, free of cost from the PDB. So this is what the beauty of data sharing here. So the reinventing of wheel approach will be significantly reduced. Okay, so once you get the data on proteins, and we can either design or we can, if you have already synthesized, we take the known actives for the LBDD. Hello, hello, yeah. So if you take both of them, you will pull the molecules, hit molecules, and whether we have to take the LBDT approach or SBDT approach. If this information is solid, we go for the SBDT. If we don't have this information, we go for the ligand-based drug discovery. So in SBDT, we have approaches like druggability prediction, okay, docking-based virtual screening approaches, pharmacophore mapping, okay, and we also have a molecular dynamic studies, and in our design we have in a structure-based drug design. Likewise, in a ligand-based drug design, we have a ligand-based pharmacophore, okay, and this pharmacophore itself is another interesting topic. Definitely, we will discuss on this in next time in our days to come. So we also have another technique called as 3D QSAR. Here, once again, we do the structure-based analysis. And we also have a 2D similarity based scaffold hopping, ADME prediction. These all are ligand based studies. Ultimately, by any means, we arrive to the hits, and these hits will be once again subjected for the in vitro assays or in vitro investigation, and it ends up with a lead molecule. And this lead molecule definitely enters the clinical trial if it is satisfactory. But what is the hurdle? I'll come to that later. So how we can reduce this effort? Okay, so how we are going to reduce the effort in identifying, okay, the positive best leads is a important point to be discussed. Let's take another example, and here we have a paper in this paper, Domen et al. This paper is published in 2002. Why I am highlighting here is this CAD is not a brand new strategy or approach. This is the oldest strategy. People are gaining much more confidence in this technique gradually. Okay, so in 2002 only, Domen et al. Have really demonstrated the strength of this computer-aided drug design or virtual screening. Why? Because you can look at the table here. They have done both docking and high throughput screening. For high throughput screening, they have taken around 4 lakh molecules. They have tested 4 lakh molecules, and the hits, number of hits they have obtained is 85 hits they have obtained with the IC50 less than 100 micromolar, right? So how many molecules they have tested? 4 lakh. Okay, out of that, 85 good. Okay, and hits with IC50 less than 10 micromolar were only 6. The hit rate here is 0.021%. But in a docking, what they have done is they have taken or they have tested only 365. Out of 365, 127 molecules have given IC50 less than 100 micromolar was 127 molecules, right? So here the hit rate was 34%. If you can get such a huge difference by adopting or in silico docking approaches, why can't you, right? So this is what exactly the strength we have associated with computer-aided drug design over a high throughput screening. And high throughput screening requires a lot of time, consumables, everything. Docking takes, okay, 1/100th time of high throughput screening. And I have another example here. We have a paper from Sawyer et al. In 2003 and Singh et al. In 2003. Why I have taken these two papers is both of them have focused on screening TGF beta kinase domain inhibitors, right? So Sawyer et al. Have used traditional high throughput screening, and Singh et al. Have used a virtual screening strategy, right? So now let's see. This Sawyer et al. Have identified the binding site of this ligand, okay, in the active pocket of TGF beta, and they have use a technique called as X-ray diffraction, and they have released this picture saying that this is how the small molecule or an inhibitor is binding to the TGF beta like this. Meanwhile, okay, Singh et al. Have used virtual screening method, and they could present this image. Okay, see, this is how the molecule, here you can see the molecule in sticks, right? So this sticks, sorry, lines. So this line diagram you are allowed to refer. So what is the difference we have? Hardly we can find 1 to 2 angstrom difference, not even that much. right? Here, if you can achieve this much of accuracy, okay, in two different methods, okay, why should we go for the highly time consuming and uh, costlier method, okay, where or you will have to take a call like whether you will go for the LB, the ligand based virtual screening, or you will go for the STS. Likewise, we have a couple of successful drugs in the market. You might have heard of this molecule called as tirofiban, okay, a fibrinogen antagonist, and it was identified in 1992 only. And we also have three different therapeutics for the treatment of HIV: saquinavir, ritonavir, and indinavir, which were identified in 2007. Okay, uh, all these are identified by using the CAD approach only, and the very successful molecule called as captopril for as a ACE inhibitor was identified in 2010, once again using the high throughput screening, virtual screening strategies. So what is this strategy is, we call this strategy as molecular docking. Here we'll have a target and ligand. How does this ligand fits well with the target will be analyzed here. So I'll take a help of this particular image. Okay, so whatever the surface you have, okay, is a protein. This rotating ligand is a small molecule. You can see as the molecule moves closer to the protein, it highlights with the red color, right? So that is how it has a complementarity. So when it gains the complementarity in terms of electrostatic interaction or steric interactions, so this will be measured in molecular docking. And in this particular diagram, in the second diagram, you can understand we try to fit in a molecule in all possible energetically favorable conformation in an active pocket of a protein. So whichever the best one will be offered to us, right? So this energy calculation will definitely help us in identifying the right molecule which will offer us a better inhibition or agonism. So with all these efforts, when we come to the lead, okay, we will not publish until and unless we reach this particular point, right? So you just imagine we have put in a lot of effort and we have investigated. By the time I come to the lead, yes, this is going to enter the clinical trial, we will face a huge problem associated with intellectual property and commercialization. So we will get to know somebody else in another part of the world has already published this, or already patented it, or already registered the license, right? So this is what exactly the real uncertainty of pharma industry is facing when it comes to the commercialization. Okay, so how to tackle it? This is a lesson we have to learn. So for this, we have different strategies, but I will discuss only two strategies for today's discussion. So we use this bioisosteres, and what are these bioisosteres are? So these are the chemical substituents with similar physical as well as chemical properties, which produce broadly similar biological properties to one another chemical. So if you take a hydrogen, it can be easily substituted by another isotype of hydrogen, okay, and fluorine. So when it comes to the hydroxyl group, it can be substituted by the amide groups, right? So this is how we can substitute. So the replacement of hydrogen atom with the fluorine, okay, at the active site of metabolic oxidation, it can once again prevent the metabolism which is going to take place at that particular. If you go for the replacement, I don't have any cases in this diagram, that's okay. So if you have any metabolic oxidation, because when it is a drug, definitely it undergoes metabolism, right? So before reaching a drug target, if it undergoes a metabolic oxidation, by then its activity will be lost, right? So if it is the case, if you want to retain the activity, but how? So the answer is bioisosteres replacement, right? If you use a fluorine here, definitely it will avoid the metabolic oxidation, so that it will travel for a long. So here we can see fluorines will be most commonly seen in these drugs, right? Likewise, if you want to improve the polarity, right? So by then we replace hydroxyl group with other bioisosteres. So likewise, we have several options, okay? So using this, we can modify the molecule. So here you can see the MIC of this particular, that is minimum inhibitory concentration of this molecule was 2 micromolar, right? So, and the solubility was very high. When you substitute this particular group, okay, with this carboxyl group with hydroxyl group, okay, its MIC is lost. Bromine with chlorine, its MIC is lost and solubility is lost. Here it was bacteriostatic in nature, but it has become bactericide. So I hope you're all familiar with bacteriostatic activity and bacteriocytic activity. It is going to arrest the growth of sorry, bacteria. Meanwhile, it is going to kill the bacteria. Okay, we are losing a MIC, but we are offering a bactericidal activity by then, which is good. The same one. So if you don't want to kill the bacteria, you just want to arrest its growth, go for it, right? So this is how we have different possibilities to tackle the IP. And the next strategy is fragment-based drug design. Here, what we do is, if you have 3 molecules, okay, which are identified as hits, but by the time you reach the lead, oh, this molecule has already got patented or published. So what we can do is take a particular fragment from each molecule, 1 micromolar, which was offering with a MIC of 1 micromolar, 3 micromolar, and 150 micromolar. Take one group fragments from everyone and make it as a 1 molecule, which can occupy, okay, which can occupy the target site. And this is what we call as a linker. So we use 3 fragments and 1 linker to deliver almost a same activity, okay, and this really helps us in breaking the IP. This is what we do in chemical modifications to break the IP. So this really works better. So with this, I conclude my discussion on how we can amplify the pace of research in drug development with the advent of bioinformatics techniques. If you have any doubts, please feel free to ask me. Thank you.
Thank you, sir, for the wonderful and very elaborative presentation. Sir, we have a question from one of the participants. Good afternoon, sir. Recently, Covishield vaccine was reported to be ineffective against COVID-19 in African gene pool. Can transcriptomic study help us identify such challenges prior to large-scale production of such vaccines?
True. It certainly helps, but the thing is, you'll have to take the sample once again from the African population, and before taking it to the next level, it is not possible, because once you come out with a vaccine, you will have to give it, and after that, if there is any differential difference in therapeutic intervention, definitely we can take. Until and unless you administer a vaccine, you will not be able to measure its response, right? So you, to understand its difference in expression, you can definitely use transcriptomics, but not prior to the exposure. I hope I answered the question, maybe partly, because I'm not a vaccine expert, to be frank. Okay, so I don't think I will be the right person to answer this question.
Okay, sir. Participants, if you have any queries, you can just unmute yourself, or post your queries in the chat box.
The voice is breaking. 10 minutes I'm coming back. Yeah. Definitely.
Sir, I hope that's all the queries. So we, on behalf of the management, staff and students of the Department of Biotechnology, the Oxford College of Engineering, we extend your very heartfelt gratitude for this elaborative and very informative presentation, sir. Thank you so much for being with us and giving such an informative talk. Thank you so much, sir.
Thank you.
Dr. Bharath, for your kind cooperation and taking the task of delivering informative talk to the faculty members who are participating in it. And we look forward for your participation in helping our faculty to find some career or a project in collaborative fashion. And students also may send their request to you because we have given permission for them also to attend. So they also may request your help in finding the project. Please help them so that we can keep
Pleasure,
the connected. Yes.
pleasure, sir, pleasure. So if you
Thank
have any